KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.com.
Hi students, so now in this video we are discussing lipids. So in previous video we have discussed amino acids information now. What are the types of amino acids? There so different types of amino acid structures and names also we have discussed, isn't it? So in this video, so we will discuss the regarding lipids. So here, so this entire information regarding lipids, and that side now we will discuss somewhat information of nucleic acids. Nucleic acids are nothing but DNA and RNA we know very well now. So somewhat introduction information I will give you here. Now let us start to discuss regarding lipids. Now lipids are generally water insoluble. Lipids are generally water insoluble. Nalla remember panikonga, lipids are generally water insoluble. Insoluble means these lipids will not be diluted in the water. That's why water insoluble. So these lipids. Okay, man. Next one, they could be simple fatty acids. So there definitely there though these lipids definitely we will be considering as simple fatty acids. So a fatty acid, so suppose if we see there no, so a fatty acid, so that may have one carboxylic group, carboxylic group, and this particular carboxyl group attached to an R group. Okay, so once you see suppose if you take one particular fatty acid, so there what is the carboxyl group it has, that one will go and binds to the R group or attached to the R group. So now here you may get one doubt, sir, R group means what is the thing we have to consider? Means R group could be R group we can be considered as a methyl group, or the CH3 group only, methyl group we will take. Next one, or maybe ethyl group, C2H5 or higher number of CH2 groups with 1 carbon to 19 carbons. Means here R group whatever may be that we can be taken, means we can take a methyl group, we can take a methyl group, or we can take a ethyl group, or we can take a higher number of CH2 groups. So CH2 groups also with 1 carbon to 19 carbons. Okay, so there try to give one example, sir, we will understand. Means once you see there, so first example, palmitic acid. Palmitic acid has 16 carbons including carboxyl carbon. So this is NEET examination point of view also even important. Okay, for example, palmitic acid, one more time try to say along with me, palmitic acid has 16 carbons including carboxyl carbon. Okay, including carboxyl carbon, palmitic acid has 16 carbons. Next one, arachidonic acid. One more time, slowly I will tell you. Arachidonic acid. One more time, arachidonic acid. Okay, arachidonic acid has 20 carbon atoms including carboxyl carbon. So means here in these two examples if we see there including carboxyl carbon we are discussing. In case of palmitic acid, 16 carbons are there including carboxyl carbon, and in case of arachidonic acid 20 carbons are there including carboxyl carbon. This is remember. So 20 carbons where we can see, So in arachidonic acid, 16 carbons in palmitic acid. Okay, first of all, so one more time let us concentrate. So lipids are generally water insoluble. So in neat examination point of view, so a lot of information not to be discussed, to be given, but here now for the first time we are learning only according to the textbook only. No? So after completion of this entire information once, so I will start neat classes. So thereby I will give elaborate information under lipids in easy mode, in easy manner. So no worry about it. Now here they could be so lipids, or they could be a simple fatty acids. So definitely the simple fatty acids definitely consist of carboxyl group, carboxyl group, and this particular carboxyl group is attached to the R group. So here the R group may be methyl group or ethyl group or higher number of CH2 groups maybe, or may have 1 carbon to 19 carbons. So that's why here we took now 1 carbon to 19 carbons. So that's why here we are discussing 2 examples. One palmitic acid. Palmitic acid, how many number of carbons are there? So 16 carbons including carboxyl carbon. Next one arachidonic acid, if you see there now 20 carbon atoms are there including carboxyl carbon. Next one fatty acids could be saturated. So fatty acids, we can be considered fatty acids could be saturated, or we will be considering into 2 categories. So one is saturated and one more is unsaturated. So I didn't underline here, one is saturated and one more is unsaturated. So fatty acids could be saturated means what is the main important thing? No, so without double bonds. So without double bonds means here the fatty acids saturated one, if you see there now their double bonds will not be there, without double bonds will be there, and unsaturated fatty acids, if you see there now with 1 or more double bonds will be there, means double bond means between the carbon, so we can observe the presence of double bonds. So thereby saturated means kandipa we can remember presence of, or there is no double bonds between the carbons. Double bonds not there means adi saturated one. Suppose 1 or more, at least 1 double bond must be there in unsaturated fatty acids. Point purida simple suppose saturated fatty acids means when we can take suppose only single bonds are there between the carbon. Ipo rendu double bonds here we draw no, suppose here we wrote no inge one irukana adi which one saturated. Without double bonds means there single bonds only no are present. Technically you try to think. Without double bonds means there kandipa single bonds only are present between the carbon to carbon. And double bonds means at least 1 or more double bonds must be there in unsaturated. Double bonds irukana unsaturated, double bond illiyana saturated. Easy? Right. Nalla save pannikonga. Next one, this definitely I will mention in your examinations. That's why whether I will mention or not, but as students we have to learn. So nalla save pannikonga. It is not pressure. If you have interest to learn, definitely you will learn this one. Right. Another simple lipid is next one more type. No. So another type is glycerol. Glycerol, which is so glycerol chemistry point of view, this particular glycerol we can be taken it as trihydroxypropane. So trihydroxypropane is nothing but glycerol. So simple point of view, so simple name point of view we can be taken this one as a glycerol, and chemistry name point of view we will be considering this one as trihydroxypropane. Next one, many lipids have both glycerol and fatty acids. So what are the lipids here we have started to learn? No, those lipids have, so once you see there both lipids have both glycerol and fatty acids. Both glycerol and fatty acids. So here the fatty acids are found esterified with the glycerol. So here we are discussing now many lipids consist of both glycerol and fatty acids. Now here in the next line now we are discussing relationship between these two, glycerol and fatty acids, or esterified. So that is only the fatty acids are found esterified with glycerol. So these two, how these two are linked together means ester bonds are there between the glycerol and fatty acids. Amma nalla save pannikonga sariya. So next one, then fatty acids are found, that is completed. Next one, they can be. So they can be then. So here these what are the glycerol and what are the lipids are there. So here once you see many lipids, both the glycerol is there as well as fatty acids are there. Fatty acids and glycerol both are linked together with ester bonds. Esterified means ester bonds, presence of ester bonds. Next one, they can be. So here these are all again these kind of lipids we can be divided into monoglycerides and diglycerides and triglycerides. Don't forget, my dear students, there glycerol is there as well as fatty acid is there. So there monoglycerides are there, diglycerides are there, and one more is triglycerides also are there in a NEET examinations point of view. So while taking NEET classes, even structures also we will discuss. Okay, no worry. So now just the types only you try to remember now. Monoglycerides are there, diglycerides are there, and one more is triglycerides are there. So these are all also called as. So one more names of these are all fats and oils. So triglyceride, actually here they must give this one as a triglycerides. So these triglycerides are also known as fats and oils. Okay, so means here in our textbook point of view they didn't mention very clearly, but triglycerides for our convenience to study here based on the melting point. So based on the melting point, triglycerides we divided into fats and oils. Ipo save pannikonga. One more time, based on the melting point, triglycerides are divided into two types. One is fats and one more is oils. Sarema. Now so here once you see there, so oils have lower melting point. So here fats and oils are there. If you take oils point of view, so these oils have lower melting point. This NEET examination point of view is important. Lower melting point oil, best example is gingerly oil, and hence remains. So because of the lower melting point, so it remains as oils even in winter also. So suppose if you see vanaspati, some kind of dalda, so if you expose to the lower temperatures, that will be like converted into jelly-like structure. That will be like a solid state, it will be converted. So but here due to the low melting point or lower melting point, oils even in winter also it will be like as like tanni madri only will be there. So that is a point here we have to understand. Next one, some lipids have phosphorus and phosphorylated organic compound in them. Here some other kind of lipids. So some other kind of lipids, actually this kind of lipids we will be considering as compound lipids. Okay, so there phosphorus will be there and phosphorylated organic compound is there. So means when the phosphorus is there, so that particular compound or that particular lipid we will be considering as a phosphorylated organic compound. So we will be considering. So this one is the best example of complex. So pataka, so thereby some lipids have phosphorus. So phosphorus and the phosphorylated organic compound in them. So these are phospholipids. So phospholipids are the best example of compound lipids. So structure also let us discuss in NEET exam. But here just oral point of view, so information point of view nalla save pannikonga. Okay, phospholipids. Next one, they are found in these kind of phospholipids, where we can see the presence of these phospholipids means, so we can found in cell membrane. So in cell membrane, the presence of phospholipids, that's why while explaining cell membrane in Cell the Unit of Life chapter, cell membrane we discussed, so there biolipid layer is there, that biolipid layer is made up of chemically phospholipids. So there best example lecithin is one of the example. So under or comes under phospholipids. Lecithin is again I should not say all the time NEET examination point of view important, but here definitely I have to mention. Lecithin is the best example of phospholipids. Generally, phospholipids helps in development of or formation of a cell membrane. Next one, some tissues, especially. So, so up to here, somewhat information regarding lipids. Okay. So, next one, if you see there now, so here some tissues, especially neural tissues. Generally, neural tissues have lipids. So here, what are the tissues are present in the neural tissue? What are the lipids are present in the neural tissues? Now have with more complex structures. So there the complexity is more. Definitely have more complex structures. Definitely they lipids only, but there somewhat a depth one. Where in neural tissues. Neural tissues are the up to here regarding lipids. Next one, somewhat another new concept will be started, that is living organisms have a number of carbon compounds in which heterocyclic rings can be formed. So some other kind, we have started to discuss some other kind of carbon compounds here. So there living organisms have a number of carbon compounds in which heterocyclic rings can be found. Heterocyclic rings we will find out. So here some of these are nitrogen bases. So here heterocyclic rings, here the best example, what is the thing we will be considering means nitrogen bases. Suppose if you have the knowledge on the DNA and RNA, I don't know. So, but here, what is the point here? What is the point here? We have to discuss. No, DNA is made up of three carbon compounds or three compounds simply. So, first one is pentose sugar, second one is nitrogen base, and third one is phosphate. Okay, so here these three compounds information in this video we will discuss. So there one of the components is nitrogen bases. There adenine, guanine, cytosine, thymine, where we can find out in DNA. So one more time. So nitrogen bases, what are the types of nitrogen bases we will find in a DNA? No, adenine, guanine, cytosine, thymine. Suppose if you take RNA, there what are the types of nitrogen bases means LMA present except this one thymine. Thymine is not present in the RNA. So instead of thymine, so we will find uracil in RNA. So thereby here one point you try to get confirmation. Nitrogen bases in a DNA are adenine, guanine, thymine, and in RNA adenine, guanine, cytosine, uracil here. So adenine, thymine is present in a DNA and uracil is present in RNA. So this is save pannikonga. Along with these nitrogen bases, when we found attached to the sugar, so once you see there, once when nitrogen bases are attached to the sugar, so which one is formed means nucleosides formation takes place. So there once you see nucleoside is nothing but, nucleoside is a new technical word, nucleoside is nothing but the combination of sugars with nitrogen bases. So thereby, suppose in slip test examination, suppose if I ask what is nucleoside means, what you have to write down? No, simply nitrogen bases plus sugar is nothing but nucleoside. Easy, one more time once you see, nucleoside is nothing but the combination of nitrogen bases plus sugar. Sugar, suppose if you like to write clearly means pentose sugar you can write. So thereby pentose sugar plus nitrogen bases together form nucleoside. Suppose for example nucleoside, suppose if you attach phosphate group also, the nucleoside, suppose if we attach phosphate group also means that will be converted into nucleotide. Okay, nucleotide. If say the nucleotide is nothing but the combination of three different types of components. What are those? What are those here? Phosphate group we are observing. Along with the phosphate group here you see the pentose sugar is there and nitrogen bases are there. So now if you take nucleotides, no, three components together forms the nucleotide. Say there First one is nitrogen bases, second one is pentose sugar, and third one is phosphate group. These three together forms the nucleotide. Suppose if I ask you to write down nucleotide means, simply you can write like this: nucleoside plus phosphate is nothing but nucleotide. Suppose if I ask to write down nucleoside means, what you will write? Nucleotide minus phosphate. Correct only no? Tide minus phosphate means nucleoside only no comes. So thereby what is the point here we are understanding? Nucleoside would phosphate will not be there. Phosphate attach avana nucleoside nucleotide avum. Amma easy only no? So one more time nalla save pannikonga, important. Next one, nucleosides. Nucleosides, once you see, what are those? Adenosine, ipo paathuka, adenosine, guanosine, thymidine, uridine, cytidine. So there here, this one we will find out in a DNA, and uridine is present in RNA. So there, what are the types of nucleosides? Total how many types are there? 5 types are there. Adenine. So here nitrogen bases, somewhere else here we have discussed adenine, guanine, cytosine, thymine. So likewise we see no. So likewise, so phosphate. 1 minute, amma. So here the nitrogen bases with the sugar no definitely forms the nucleosides no. So there sugar plus nitrogen bases 1. So here adenine with sugar no adenosine, and guanine with sugar guanosine, and thymine with sugar or pentose sugar thymidine. Next one, pentose sugar with uracil uridine, and cytosine with pentose sugar cytidine. Likewise nucleosides formation we can observe. Next one, nucleotides means, so what are the examples no? So here nucleotide means now only we learn the combination of 3 different types of components. There phosphate is there, along with the phosphate, pentose sugar and nitrogen bases, isn't it? Now adenylate acid is there, thymidyllic acid is there, guanylic acid is there. Means here, did you understand one point? Acid attached. Adenine ki pentose sugar add avuna adenylic adenosine vandachi. Phosphate also attach avuna adenylic acid achi. Point you understood ma? Ipo adenine is there, nitrogen base. Ipo here you see adenine is there. For adenine, when pentose sugar is attached no, adenine is now we are calling as adenosine. Ipo phosphate also is attached means adenylic acid. Amma point purida? So thereby here what is the point here we are understanding no? Nucleosides means so here adenosine plus pentose sugar converted into adenosine, likewise all these things. Next one, nucleotides point of view: adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid. Likewise acid comes means that you try to remember it as tide, nucleotide. Next one, these 2 points easy only. Now nucleic acids like DNA and RNA consist of nucleotides only. So here we are discussing nucleosides and nucleotides. So sorry, these 2 things, these 2 statements we are discussing no. Out of these 2, which one is present in nucleic acids? Sir, means in nucleic acids means in a DNA and RNA kandipa we will observe the presence of nucleotides. Okay, nucleotides. So thereby here what is the point now? In nucleic acids we will observe the presence of only nucleotides, means the combination of 3 different types of components. Don't forget, nucleotide is nothing but presence of 3 different types of components together. Next one, so here the DNA and RNA acting as genetic materials. So here most of the organisms, if you take there no, in most of the organisms DNA acting as a genetic material, and in most of the viruses, or simply you try to remember it as in viruses RNA is genetic material. Okay, so here these 2 points you try to say. So here when you will come to the second year no, so at the time some more information, detailed information I will give you both DNA and RNA. So very detailed information we will discuss in +2 studies. Okay, now here examples point of view, somewhat diagrams we have to draw and we have to learn also. Okay, now here the nitrogen bases point of view. So nitrogen bases, here I didn't give somewhat information related to the nitrogen bases. So here for our convenience to study nitrogen bases, we are dividing into two types. One is a purine and the second one is pyrimidine. Okay. Among nitrogen bases, two types of nitrogen bases are there, purine and pyrimidines. If purines, so what are the types? If here we see no nitrogen bases, adenine, guanine, cytosine, thymine. Likewise we see no, if these four types, now let us consider into, or let us divide it into two types, purine and pyrimidine. If purine, so what are the purines are there means adenine and guanine. So try to write down carefully. Adenine and guanine, these two are purines. Next one, pyrimidines are cytosine thymine, and here one more uracil. So here these two cytosine and thymine are present. Thymine already we see no, thymine only is present in DNA, so thereby cytosine, thymine is present in DNA, and cytosine, uracil present in RNA. Okay, so thereby generally if you take a pyrimidines, no, three we have to take, cytosine, thymine and uracil. Okay, so these three are pyrimidines. Okay, so thereby if a purine structure if you see there, no, so these purines are dicyclic compounds. Purines are dicyclic compounds. So di means two cyclic compounds present together. Once you see this one particular cyclic compound and pakatali this one more cyclic. So that's what this one cyclic, that's why purines are dicyclic compounds, and pyrimidines are monocyclic compounds. This one mattandana irukku, this monocyclic compound. Pakatali vere irukana, again this one also will be dicyclic, but pakatali vere not there now. So that's why this monocyclic compound. Amma point puridana. Purines are adenine and guanine purines, and pyrimidines are cytosine, thymine, uracil. But don't forget one point, thymine is present in DNA and uracil is present in RNA. I hope you understood. Suppose if you are unable to understand, no worry my dear students, your teacher is there in front of you. Okay, man, definitely you learn this entire information, so definitely it will be there in your mind before going to the examination. Okay, NEET examination. Definitely I'll give that particular assurance to you. Okay, next one, nucleoside means, once you close your eyes and nucleoside is nothing but the combinations of phosphate, nucleoside is nothing but nitrogen bases with pentose sugar, isn't it? But there in a nucleoside, which one is absent? Phosphate is absent. Correct only, no? Now once you see nucleoside means here this particular structure is a pentose sugar. Now particular pentose sugar first carbon attached with adenine. See there. Here totally this particular structure, why we are calling as pentose sugar? No, pentose means 5 carbons are present here, 1, 2, 3, 4 and this is 5th. One more time, here 1 carbon irukama, inge second one, inge ulle third one, inge fourth one, inge this is 5th one. So there this first carbon we took, inge first carbon alli this adenine attached, no. So that's why here this particular structure we will be considering as adenosine. Okay, now pakatali par inge this nucleotide. Nucleotide means phosphate is attached newly, no. So ange 5th carbon ki CH2OH is there, no. On the side CH2OH, OH generally is there means adi highly reactive, no. That will not be like a stable. Okay, so thereby here CH2OH inge or H inge OH. So here if you see there, no, phosphate structure. Aman, let me explain once phosphate structure. Phosphate structure will be like this. P will be there center, male double bond with oxygen, 3 OH groups are there. Inge one, and here generally one more OH will be there. But here what is happening, here one OH, ange CH2OH nalli one more H is there, no, rendu may react ayi water tanni release. So that's why here this phosphate structure is attached to the 5th carbon of pentose sugar. So now here again here adenosine is there. Adenine is there, no. So now here adenine nitrogen base, 1 pentose sugar and here 1 phosphate, total together form the 1 nucleotide. So that's why here we have written this one as adenylate acid. So you see there nucleotides, adenylate acid here we took now, that particular structure here we have discussed. So idi suppose if you are unable to understand, at least practice this at home. I will not ask you this one, but once you practice, okay ma, avladha. So in this particular class no simple informations only we have discussed, no worry. So regarding lipids we discussed, and somewhat introduction information of nucleoside and nucleotides and nucleic acids information only we have discussed. So thank you and be safe at home.